How does randomization work in cancer clinical trials? Imagine you are part of a study testing a new treatment for lymphoma. You might wonder how researchers decide who gets the new therapy and who receives the standard treatment. That's where randomization comes in. Randomization is a process that assigns participants to different treatment groups purely by chance, not by choice or doctor preference. This method helps ensure that each person has an equal chance of being in either group, which is crucial for fair and accurate results. The main goal of randomization is to prevent bias. If doctors could choose who gets which treatment, they might unintentionally favor one group over another, skewing the results. Randomization also helps balance out factors like age, disease stage, or other health conditions across all groups. This way, differences in outcomes are more likely due to the treatments themselves, not other variables. There are different ways to randomize participants. The simplest is called simple randomization, similar to flipping a coin. Each person has an equal chance of being assigned to either the new treatment or the standard one. While easy to do, this method can sometimes lead to uneven group sizes, especially in smaller trials. Another approach is called block randomization. Here, participants are grouped into blocks, and within each block, they are assigned to different treatments. This ensures that the number of people in each group stays balanced throughout the trial. It's especially helpful when the study is small or when researchers want to keep groups similar in size. Stratified randomization takes things a step further. Before assigning treatments, researchers divide participants into subgroups based on important factors like disease stage or age. Randomization then occurs within each subgroup, helping to evenly distribute these characteristics across all treatment groups. This method makes comparisons more reliable because it accounts for key differences among participants. There is also adaptive randomization, which is more advanced. It adjusts how participants are assigned based on the results collected so far. For example, if one treatment appears to be working better, more participants might be assigned to that group as the trial continues. This approach can make studies more efficient and ethical but requires careful planning and analysis. In lymphoma clinical trials, randomization is especially important because patients can have very different disease types and stages. By randomly assigning treatments, researchers can fairly compare new therapies with existing options. This helps determine if emerging treatments are safe and effective, without bias. Most often, randomization is done using computer programs that generate unpredictable assignment sequences. This prevents anyone from guessing or manipulating who gets which treatment. Sometimes, trials use unequal ratios, like assigning twice as many people to the new treatment, to gather more data on that option. But this can make analysis more complex and usually requires a larger number of participants. Ethical oversight is essential to ensure that patients understand the randomization process and that no one is unfairly disadvantaged. Regardless of the group they are in, all patients receive the same standard of care and monitoring. In summary, randomization is a vital part of cancer clinical trials, including those for non-Hodgkin lymphoma. It helps eliminate bias, balances participant characteristics, and provides trustworthy data about new treatments. This process is fundamental to advancing lymphoma care safely and effectively, always with the goal of improving patient outcomes. If you are considering participating in a trial, always discuss with your healthcare provider to understand how randomization might affect your treatment options.